Hello everybody, this is Dr. Nadeem and we are with Neelam Path Lectures, the Pursue series. As you are aware, most of our lectures, in fact all the lectures are available on YouTube, which is easily downloadable. You can view them at your convenience. We also have a Telegram group which you can join. All the lecture related information is available here and all the various flyers of the lectures are put up here. And this is very helpful in accessing all lecture related information. We also have a Google Drive where the PDF of all the lectures are available, and these are the disclaimers, and this is phase 3, and in phase 3 we have Pursue 21E, which is immune disorders. This is the first session of immune disorders, and we are streaming from AIIMS Bhubaneswar. And to talk on the first session on immune disorders, which is the normal immune response, we have Dr. Amit Kumar Adhya. He is an MD from PGIMER-Chandigarh. Presently, he is a professor in the Department of Pathology and Lab Medicine, AIIMS Bhubaneswar. He's also been the ex-professor of Kalinga Institute of Medical Sciences, Bhubaneswar. He's got more than 118 publications in international and national journals, 1 book chapter, 6 funded projects, and 21 years of teaching and research experience. He has completed 5 collaborative research projects funded by DBT and BRNS. Was awarded the Best Paper Award at the IAPM 2005. With this, let me request Dr. Amit Kumar Adhya to start the fundamental lecture on immunology, that is normal immune response. Over to you, Dr. Amit. Thank you so much.
Hello students. In this video, I shall talk about the normal immune response. This will form the basis of understanding the immune disorders. We all know that there are 2 types of immune responses, the innate immune response and the adaptive immune response. The innate immune response is something which is inherent to our body. It is already there when we are born. This does not require any prior exposure to antigens, whereas the adaptive immune system requires exposure to antigens for activation. The components of innate immune system are the epithelial barriers, which do not allow the microorganisms to enter our body, plus the phagocytic cells such as neutrophils, monocytes and dendritic cells, along with a special type of cell known as the natural killer cell or the NK cell, and the complement system. This immune response mounts very quickly against microorganisms. The onset is within 1 hour. Hence it is known as the primary immune response. However, this immune response does not have any memory and it is not specific to any particular organism. On the other hand, the adaptive immune response takes a bit of time, that is 2 hours to 24 hours, to mount a response against a microorganism. This is also known as the secondary type of immune response. But once it recognizes an antigen, it remembers it, so memory is present, and this immune response is very specific. A particular organism activates a particular group of lymphocytes. There are only 2 receptors on the lymphocytes, namely the B receptors and the T receptors, which recognizes about 1 lakh different type of antigens. The cells of this immune system are basically the lymphocytes known as the B lymphocytes and the T lymphocytes. The innate immune system recognizes the microbes by the help of certain proteins known as the PAMPs or the pathogen associated molecular patterns. Not only the microbes, this kind of immune response recognizes dead tissue in our body also by the help of certain proteins known as DAMPs or the damage associated molecular patterns. So whenever there is an infection by a microorganism, or if some cells are dying in the body, then the innate immune system will be activated and there would be inflammation. That is why you see inflammation against necrosis. The cells of the innate immune system recognize the microbes or the necrotic tissue by the help of certain receptors which are present on their cell surface. You have learnt already about these kind of receptors in the chapter of inflammation. The most important receptors which are present on these cells are the toll-like receptors, TLRs, the node-like receptors, the rig-like receptors, and the C-lectin type receptors. I will not go into the details of all this, but one thing that is important for our purpose are the node-like receptors, which forms a complex known as the inflammasome. Sometimes inflammasome is asked as a short note, so please pay attention here. The inflammasome is basically a complex which is formed by three things: the node-like receptors, an adapter molecule, and caspase, as seen on the figure on the right side. This complex leads to activation of caspase 1. The caspase 1 is an enzyme or a protease which cleaves pro-interleukin 1 beta into interleukin 1 beta, and this active interleukin 1 is secreted into the circulation, which brings about severe degree of acute inflammation. A mutation in node-like receptors is also known to cause a disease known as autoinflammatory syndrome. This is different from the autoimmune disorders that we will learn in subsequent lectures. This is also associated with the pathogenesis of certain diseases like the gout, the type 2 diabetes, and atherosclerosis. We'll again read about these inflammasomes and its association with atherosclerosis and type 2 diabetes later on. Gout, as you know, is a disease which is caused due to deposition of calcium crystals or within the joints or in the soft tissue. That leads to formation of inflammasome and inflammation in that area. Now coming to the adaptive type of immune system. As I have already mentioned, the adaptive immune response consists of two cells, which are the lymphocytes, the T cells, and the B cells. The B cells produce plasma cells in response to antigenic stimulation, and the plasma cells produce antibodies. So they mediate what is known as a humoral immunity. On the other hand, the T cell mediate cell-mediated immunity. There are various types of T cells, three of which are very important for us. The helper type of T cell, which contains CD4 on their surface. The cytotoxic T cells, which contain the CD8 molecule on their surface. And there is another subset of T cell known as the T regulatory cells, also known as T regs. These T regs, they contain CD4 on their surface, but in addition to CD4, there is another molecule known as CD25. All these T cells, they have a receptor on their surface known as the T cell receptor. The T cell receptor recognizes the major histocompatibility complex or the MHC molecules. We'll talk about TCR and the MHC molecules in detail. Let me give you an overview of the different functions of different type of cells of the adaptive immune system. The B cells, they are armed with antibodies on their surface. They recognize extracellular microorganisms. When they do so, they produce plasma cells, and plasma cells produce antibodies. As you can see in this figure, the antibodies can bind to the microorganisms, thus causing their neutralization. Or the antibodies can bind to microorganisms and make them tastier for the macrophages, which is known as the process of opsonization and phagocytosis. The macrophage then eats up the microorganism and kills it. Or they can lead to activation of the complement system. The complement system will lead to destruction of the microorganism. So important thing here to remember is that B cells are mostly active against extracellular microorganisms. On the other hand, T cells mostly respond to intracellular microorganisms. The T cells cannot recognize freely moving microorganisms. For recognition, they need antigen presentation by macrophages. The macrophages are known as antigen presenting cells because they eat up the antigen, process it, and they display it on their surface by the help of MHC molecules. These MHC molecules are identified by the T cell receptors which are present on the T cell surface. Once the MHC molecule engage with the T cell receptors, the T cells get activated. Upon activation, they produce a lot of cytokines which can lead to activation of the macrophage. They can recruit inflammatory cells such as neutrophils, and neutrophils will cause inflammation, or they lead to proliferation and differentiation of other T and B cells. In fact, the helper T cells are known as the master of the symphony because they orchestrate a lot of different immune functions and they bring in different types of immune cells to the site of injury. The cytotoxic T lymphocytes, they also recognize antigens which are within the cell. The cells, mostly the antigen presenting cells, they present the antigen or display the antigen on their surface with the help of MHC molecules, and the cytotoxic T cells recognize them by the help of their T cell receptor. On engagement of TCR with the MHC molecule, the macrophage gets activated and it kills the intracellular organism or itself gets killed. Hence the name cytotoxic. The T lymphocytes are toxic for the macrophages. On the other hand, if you see, the regulatory T lymphocytes or the Tregs, they do not kill microorganisms, rather they suppress the immune response. So they act as checkers. They check the excess immune response that might occur. If they are absent, an autoimmune or an exaggerated immune response can result. Natural killer cells, it is a special type of cell which belongs to the both to the adaptive as well as innate immune system. As the name suggests, they are natural killers. When they recognize the antigen, mostly the intracellular antigen, they will lead to killing of the infected cell. We'll talk about natural killer cell later. Here is a table which enumerates the differences between the humoral and the cell-mediated immunity. The humoral immunity is brought about by B cells and they are mediated by production of antibodies. It mostly recognizes extracellular microbes, although intracellular microbes sometimes can also be recognized. And major action by producing antibody, they cause neutralization, opsonization, phagocytosis or complement activation. On the other hand, cell-mediated immunity is brought about by helper T cells or the cytotoxic T cells, and they are regulated by the Tregs. They recognize mostly the intracellular microorganisms and their action is mostly by killing the infected cells directly. The T cell receptor. Understanding the TCR is a key point in understanding the different immune mechanisms and understanding autoimmunity. So please pay attention. The T cell receptors are present on all the T cells. As you can see in this picture, this particular, it is the T cell and this is the antigen presenting cell over here. Let us focus on the T cell. This portion is the T cell receptor. The T cell receptor consists of two chains or two proteins, the alpha and beta, which are bound together by a disulfide bond. The groove between the two alpha and beta chain forms the antigen recognition site, and mostly it recognizes the peptide antigens. The polysaccharide and glucose type of antigens are not recognized. In addition to the T cell receptor, the molecules are also associated with another protein known as the CD3 protein. The CD3 protein, as you can see, consists of three chains: the epsilon chain, gamma chain, and the delta chain. The CD3 is present on all types of T cells, irrespective of whether they are helper T cells or cytotoxic T cells or Tregs. So it is CD3 is universal T cell marker. Sometimes it is asked, what is a pan T cell marker? Pan T cell marker is CD3. In case of helper T cell, this complex is associated with CD4. But in case of CD8 or the cytotoxic T cell, this complex is associated with CD8 instead of CD4. Similarly, in Tregs, you will see CD4 as well as CD25. The CD3 acts as co-receptor. The CD4 or CD8 act as co-receptors. They transmit the signal. As you can see in this picture, the complex which is formed by the T cell receptor and CD3 transmits signal upon binding with the antigen. But there is another signal or signal 2 which is also produced by the help of this molecule known as CD28. This molecule is known as a co-stimulatory molecule. I have already mentioned that this is the antigen presenting cell. So this molecule is the MHC molecule. CD4 cells can only recognize antigens which are bound to MHC class 2. So they are known as MHC class 2 restricted cells. Whereas the cytotoxic T cell or the CD8 positive T cell recognize the antigen when they are bound to MHC class 1. So they are known as class 1 MHC restricted molecule. CD28 is present both in the CD4 and CD8 positive cells, and they are known as co-stimulatory molecule. The importance of co-stimulatory molecule is very high because this second signal is necessary for activation of the T cell. If the T cell recognizes an antigen in the absence of this signal co-stimulatory molecule, or if the co-stimulatory molecule is not engaged to the CD80 which is present on the antigen presenting cell, that leads to a condition known as anergy. A-N-E-R-G-Y, anergy. Anergy is a condition where the T cells upon binding to the antigen becomes totally inactive. So it cannot mount a response against the particular antigen. This energy is very important term, and you will understand its importance when we talk about autoimmune disorders of the body. I have already mentioned that the T cell receptor consists of two proteins, the alpha protein and the beta protein. Most of the T cells will have this alpha and beta protein, but there is another subset of T cells which will have gamma and delta protein instead of alpha beta. So according to the type of protein, T cell receptors can be alpha beta type or gamma delta type. In fact, most of the T cells will have alpha beta type of T receptor, and this alpha beta type of T receptor can only recognize antigen when it is bound to MHC molecule. So they are MHC restricted. But the gamma delta T cell receptors, they do not need MHC molecule for recognition of antigen. This gamma beta type of T cells are usually present in the mucosal surfaces, like in the Peyer's patches of the GI tract or the tonsils. An important thing about this gamma delta TCR is that this kind of T cells can give rise to lymphomas or malignant neoplasm. In fact, any T cell can give rise to malignant neoplasm. Gamma delta TCRs having T cells, they produce lymphomas at the mucosal sites. Similar to the T cell receptor, the B cells also contain B cell receptors. The B cell receptor is basically much simpler than the T cell receptor, as you can see here. This whole thing is one B cell receptor, which is composed of an immunoglobulin. We all know that immunoglobulin have two light and two heavy chains. In this picture, the immunoglobulin is IgM type. You can have different types of immunoglobulins, IgG, D, A and E type. This B cell receptor, particularly shown in this picture, has a IgM type antibody on its surface, and it is associated with some other proteins such as the immunoglobulin beta and the immunoglobulin alpha. These are the heavy chains, these are not the complete immunoglobulins. There is another molecule, the CD21. I hope you know what is the full form of CD. CD means cluster of differentiation. These are different types of proteins which are present on the surface of different types of cells, and they help us in identification of the type of cell. They are known as markers. So if something is CD4 positive, that means that cell is a helper T cell. So CD4 is a marker of helper T cell. Similarly, CD8 is a marker of cytotoxic T cell. And all T cells have CD3, so CD3 is a marker of all T cells, irrespective of whether that is CD4 or CD8. On the other hand, the marker for a B cell are CD19, CD20 and CD79A. This immunoglobulin alpha is basically what is a CD79A, and immunoglobulin beta is CD79B. These two molecules are very essential because they act as signal transducers. Like TCR, here also two signals are sent. One signal is sent from the complex which is formed by the B cell receptor and CD79A and B, and another signal is sent by engagement of the antigen with CD21. Now, interesting fact here is that CD21 acts as a receptor for the virus called Epstein-Barr virus. So absence of CD21 makes the person immune to infection by Epstein-Barr virus. Another cell of importance to us is the dendritic cell. As you can see in this picture, the cell has got numerous dendrite-like processes arising from the cell surface, hence the name dendritic cell. In fact, this is a macrophage. This is a special type of macrophage, and macrophages are derived from the monocytes of the blood that you already know. These cells, the primary function is to present the antigen, that is, they will eat up the microbe, break it down, display them on their surface with the help of MHC molecules, and the microbial antigens will be shown to the lymphocytes. So the name antigen presenting cell. They present the antigen to other immune cells. They are usually located in the skin and mucosa or in the lymph node, because these are the sites from where the microorganisms gain entry to our body. At that site, the microorganisms are captured by the dendritic cells and they are carried to the lymph node, where they present it to the lymphocytes present within the lymph node, and once the lymphocytes recognize that this is a foreign antigen, they will mount an immune response. The dendritic cells which are present in the skin are also known as Langerhans cell. This is different from the Langhans cell which you see in tuberculosis. This is Langerhans and not to be confused with Langhans. In the lymph node, the dendritic cells are either present in the interfollicular regions, where they are known as the interfollicular dendritic cells, or they can be present within the follicles, which are known as the follicular dendritic cell. Their main function is T cell activation, but the follicular dendritic cells can lead to B cell activation as well. Again, the marker for this cell is CD1A and Langerin. The natural killer cells. These are a very important subset of cells present in our body. They are mostly the cells of the innate immunity, but they also act with the adaptive immune cells. The major function is to destroy irreversibly stressed and abnormal cells in the body, such as the virus infected cells and tumor cells. Recently, the role of NK cells in killing of tumor cells has been recognized, and vaccines are being experimentally tried with the help of these NK cells to kill tumor. The NK cell contains two molecules on their surface known as CD16 and CD56, which also serve as the markers for NK cell. I am always stressing upon the markers because these markers are important MCQ questions. The CD16 is a Fc receptor and it mediates a type of immune response called as ADCC, antibody dependent cell mediated cytotoxicity. We'll learn more about it gradually in the later subsequent videos. But for now, the NK cell has got two actions, or as you can see in this picture, the NK cells have got two receptors on their surface. One is an activating receptor, another is an inhibitory receptor. The NK cells keep circulating in our body and they are the cells of surveillance. They keep a surveillance or track. They will check every cell in the body whether it's normal or abnormal. If they encounter a normal cell, then the normal cell will only engage with the inhibitory receptor and hence there will be no action. The NK cell will be inhibited, it won't kill the cell. But if the cell is abnormal, such as a tumor cell or a cell which is infected by a virus, then the cell usually engages with the activating receptor, thus activating the natural killer cell, and as the name suggests, it will lead to killing of the cell. This is in short the function of NK cell. We will be talking more about NK cells later. Another important thing that you must know are the tissue of the immune system. There are two types of tissues, the central tissue and the peripheral tissue. The central tissue basically refers to bone marrow and thymus, where the immune cells are born. Whereas the peripheral tissue, also known as the secondary lymphoid organs, are basically the lymph nodes, spleen, the mucosa associated lymphoid tissue and the gut associated lymphoid tissue. The right hand side shows a picture of a lymph node, a cut section of a lymph node. you know that the lymph node has got a capsule, and beneath the capsule there is a subcapsular sinus. The outermost portion of the lymph node is known as the cortex. The area which is near the hilum is known as the medulla, and the area in between is known as the paracortex. The cortex contains mostly B cells, the paracortex contains mostly T cells, and the medulla contains mostly lymphocytes and plasma cells. There are some afferent lymphatic vessels through which the lymph enters into the lymph node. These afferent lymphatic vessels enter the lymph node through the capsule and drain into the subcapsular sinus, whereas the efferent vessels, they come out from the hilum. The artery enters the lymph node through the hilum, and the vein comes out from the lymph node through the hilum. So only the afferent vessels, they do not enter through the hilum, rather they enter through the subcapsular sinus. This is important because the lymph drains from various areas, and thereby the antigen presenting cells, mostly the dendritic cells, also enter into the lymph node via the afferent lymphatic vessel. They go into the follicular region where the follicular dendritic cells are sitting. They also move into the interfollicular region where the interfollicular dendritic cells are there. So they present their antigen to these dendritic cells and lead to activation of the lymphoid cells. Major histocompatibility complex. This is another very important concept that all of you must know. In fact, in your syllabus there are few things which are labeled as must know. There are few things which are labeled as desirable to know, and few things which you may or may not know. For passing, you have to know everything that is under the must know category, and MHC molecule is one such thing which is a must know for you. For MCQs, mostly the desirable to know are the things which come as MCQs. So you are left with no option, you need to know everything basically. The major histocompatibility complex, also known as the human leukocyte antigen. The major function of this molecule is to display the peptide fragment of protein antigens for recognition by antigen specific T cells. I repeat, the major function of MHC molecule is that it is present on the surface of the cells, and it helps in displaying the antigens, mostly the protein antigens on their surface, which are then recognized by T cells, and then an immune response is activated. So you can understand what is the importance of this MHC molecule. In the absence of MHC molecule, basically there would be no adaptive immune response. The gene which transcribes for the MHC molecule is located on the chromosome number 6, and there are three classes of MHC molecules: the MHC molecule 1, class 2 and class 3. If we look at this diagram, this shows the gene for the MHC molecule. The class 1 MHC molecule basically has got three loci, the locus A, B and C, whereas the class 2 MHC molecule has got the D locus only. The D locus, however, has got three different areas: the DP, DQ and DR. In addition, in between these two classes there is another class 3 molecule which mostly is associated with production of Sorry for the interruption. So as you can see here, the MHC genes, they produce the MHC molecules. The class 1 MHC molecules consist of two fragments, the alpha protein and the beta 2 microglobulin. The alpha chain is again divided into the alpha 1 domain, alpha 2 domain and the alpha 3 domain. The groove between the alpha 1 and alpha 2 domain forms as the site of peptide binding. The alpha 1 and alpha 2 domains are very variable and they are quite polymorphic. No two MHC molecules in two individuals would be the same. On the other hand, the alpha 3 portion is constant or fixed and it is same for all individuals, and this is the site for recognition of the CD8 molecule. On the other hand, the class 2 MHC molecule has got two proteins, the alpha chain and the beta chain, and the groove formed between the alpha and beta chain forms the site of peptide recognition. It does not have the beta 2 microglobulin associated with it. Here is a table which enumerates the differences, the functional differences basically. In the previous slide we saw the structural difference and the difference at the genetic level of the class 1 and class 2 MHC molecules. This table tells you about the functional differences. The most important difference is that class 1 molecule is present on all nucleated cells of the body, and usually it displays intracellular protein antigen such as the virus and the tumor antigen. On the other hand, class 2 molecules are only present on macrophages, B cells and dendritic cells. That means it is mostly present on the antigen presenting cells and the B lymphocyte, and it deals with mostly extracellular microbes and soluble proteins. Class 2 molecule, as we all know, is recognized only by class CD4 positive T cells or the helper T cells. On the other hand, class 1 molecule is only recognized by CD8 positive T cells or the cytotoxic T cells. Let us now understand the overall mechanism of the cell-mediated immunity. As you can see in this picture, the dendritic cells or the antigen presenting cells, they capture the antigen from the mucosal sites or the site of injury, and then they move into the lymph node via the efferent lymphatics. Within the lymph node, they present the antigen to the CD4 positive T cells as well as the CD8 positive T cells. Upon binding, upon recognition of the antigen, both this type of subset of T cells, they proliferate and they produce what is known as the effector T cells. The effector T cells then move into the circulation and reach the site of injury. At the site of injury, they again bind with the antigen presenting cells or the phagocytes and then bring about their immune response. The CD4 positive T cells recruit inflammatory cells causing inflammation, or activate the phagocytic cells, and the macrophage leads to killing of the microbe by the process of phagocytosis. On the other hand, the cytotoxic T cell will activate the infected cell and the infected cell will die, and hence the immune response is brought about. Also note that the naive T cells, when they are exposed to the antigen, they produce interleukin 2. A naive T cell is a cell which has never encountered an antigen. For the first time it encounters an antigen and produces interleukin 2. The interleukin 2 acts in an autocrine manner, that is, it is produced by itself and it acts on itself. A paracrine is something which is produced by one cell and it acts on other cell, that is paracrine. This interleukin 2 leads to proliferation of the T cells, which then differentiate into the effector T cells and the memory T cells. The memory T cells remain circulating in the blood, and the effector T cells, they go to the site of inflammation and injury, and then they cause the immune response. The T helper response is a very important thing. In fact, there are three different type of helper responses: the Th1 type response, Th2 type response, and Th17 type response. This table nicely depicts the differences between the three responses. Sometimes it is asked as question, the difference between Th1 and Th2, or the difference between Th1 and Th17. You need to go through this table yourself. I will highlight the most important things here. You see that Th1 response is mostly directed towards intracellular microbes, whereas the Th2 response is mostly mediated by immunoglobulin E by the cell's mast cells and eosinophils, and it acts against helminthic parasites, and it is responsible for causing allergies or type 1 hypersensitivity. We'll again talk about this when we discuss type 1 hypersensitivity. On the other hand, the Th17 type of response is mostly against extracellular bacteria or fungi, and this is the major cause of autoimmune diseases such as inflammatory bowel disease or psoriasis. Please go through the table in detail. The humoral response, as we all know, is brought about the B cells. The B cells can recognize extracellular microbes. The naive B cell, or the B cells which have never encountered an antigen, are usually armed with IgM or IgD type of immunoglobulin on their surface. When a microbe attaches to these, if you remember, this also forms the B cell receptor. The same microbe also attaches to helper T cells, and helper T cells lead to proliferation of B cell. Upon proliferation, a subset of the B cell differentiates into plasma cells which produce immunoglobulins. In the early stages, immunoglobulin M is produced. Later on, there is a class switch. That is the immunoglobulin M producing B cells. Now they start producing immunoglobulin G. So in the later stages of infection, you find a lot of immunoglobulin G in the serum instead of immunoglobulin M. On the other hand, some subset of the B cells, they become memory B cells. The immunoglobulins that are produced by the B cells, they will lead to various type of responses, such as either neutralization of the microbe or opsonization and phagocytosis, or they will lead to activation of NK cells, thereby causing antibody dependent cytotoxicity or ADCC. Please remember this, ADCC is brought about by NK cells by engagement with the immunoglobulin. You know that the immunoglobulin has two ends. One is the antigen binding end or the Fab end, and there is an FC end. The FC end acts as a binding site, and the receptor for FC are present on many cells in our body, including the NK cells and most of the antigen presenting cells. The antibodies can also lead to the antigen antibody binding, usually leads to complement activation, and complement by its own mechanism kills the cells. Complement activation also leads to production of anaphylotoxins such as C3A. It also leads to formation of opsonins such as the C4B, and it also leads to production of the membrane pore formation complex or the membrane attack complex. This was an overview of the normal immune response in our body. This will form the basis to understand the immune mediated diseases, which I will be taking in the subsequent lectures. Thank you.